This is the video for Chem 223, Lecture 17. Today we will be talking about chromatography fundamentals. So before we get into what those fundamentals are, let me give you an example of a problem that we've used chromatography to investigate or solve. So this is a real-life problem that some people in the life sciences building are working on at BYU. And that is asking whether or not certain things are happening to fish in Utah Lake. Now, Utah Lake has several fish species. I've just got a few of them listed up here on the slide. Bluegill, by the way, are really easy to catch. They're not very smart, so you can just drop a hook in without any bait. Usually catch a couple of those. But the problem is that in Utah County, where BYU is, a lot of people take antidepressants for a various number of reasons. And those antidepressants come out in urine or other waste, and they come out in the toilet, and then they get flushed down into Utah Lake. So the question is, how do these antidepressants affect the fish? So the way that these researchers are going to try and answer this question is they're going to catch a bunch of fish out of Utah Lake. They're going to put them in aquariums, and in some of the aquariums, they're going to just put clean water in. And in other aquariums, they're going to put water with antidepressants at the same concentrations as are found in Utah Lake. But before they can do that, before they can dope up these fish with antidepressants and make them feel better, they first have to figure out which antidepressants are present and at what level. So they're working on a two-part problem. They're first trying to qualitatively analyze these samples of lake water and figure out which antidepressants are present, and then they want to quantify them. So this is a qualitative and quantitative experiment. And what they're going to do for the determination of what antidepressants are in the lake and at what concentrations is they're going to use a technique called chromatography. So chromatography is a separation technique, meaning that we use it to try and separate chemicals as completely as possible, including when you have hundreds or thousands of chemical species in a sample all at once. And that is almost always the case when you are dealing with a biological sample, which is what we do in my lab all the time with chromatography, is try and separate out protein and peptide samples that have thousands of proteins and peptides in them. Right, but chromatography is Greek for color right. The reason it's called that is because it was first used in the year 1900 by Mikhail Svet to separate plant pigments. And whenever I see a number like this, 1900, I feel suspicious. Why is it such a perfectly rounded number? But it was in 1900 when Mikhail Svet used it to separate plant pigments. And the way chromatography works is it uses either chemical or physical properties to separate your mixtures. And like I said a little while ago, the goal is to separate components as completely as possible and hopefully as quickly as possible. Sometimes this can take 4 or 5 hours or even longer, depending on what species you're trying to separate. And so complete separation is usually the overall goal, but reducing the amount of experimental time is also really important, especially when you're trying to do hundreds of separations. There's 5 main types of chromatography. We're just going to introduce each of them really briefly today, and that'll be our lecture. The first type is adsorption, then second is partition, third is ion exchange, fourth is size or molecular exclusion, and fifth is affinity. Now, before we get into the specifics of these different types of chromatography, I want to introduce you to some chromatography vocabulary, and this will be useful for your understanding of how chromatography works. The first phrase that you need to learn is chromatography column. This is some piece of tubing that contains what we call a stationary phase, and it allows a mobile phase, such as a liquid or gas, to flow through it. Okay, now the mobile phase in these experiments is a solvent or gas that moves through the column and carries your analyte through the column. So if you have a sample of lipids and you're trying to separate out the lipids, you would inject those lipids into the mobile phase, and the mobile phase would carry it through the column, and while they move through the column, they'll interact with your stationary phase. The stationary phase is a phase or substance that just stays put in your column. So as your analyte is passing through the column over your stationary phase, it'll be interacting with it, but the stationary phase won't be moving. Right. The next term that I'm going to introduce is elution. And this is the process of passing a liquid or gas through a chromatography column. And then the last phrases we'll cover are packed and open tubular columns. So these are different types of columns or different ways of preparing your columns. So a packed column is filled with particles containing the stationary phase, and an open tubular column is narrow. There's a narrow capillary with a hollow center, and the stationary phase is coated on the inside of the walls. Give you kind of a graphical representation of what these would look like. This is a representation of a packed column. You'll notice it's got a bunch of circles or beads inside of it. It's because in a packed column, we usually actually pack it with little tiny beads. If you can see this white bar right here, that corresponds to 1.5 micrometers or 1.5 microns. So these beads that are in here are really small. This can often just look like a powder or feel like a grainy substance. Usually actually pretty fun to feel with your fingers, so they usually feel pretty cool. But these beads are really tiny, and then the surface is coated with something that we want our analyte to interact with, at least briefly, as it passes through here. Now, open tubular columns can look like one of these two examples here. So you can actually have beads stuck to the walls of your column and have the interior empty. And you can do this a lot of times for gas phase chromatography, where you're just pushing a gas through the column, or for various other reasons. And then you could also, instead of having your stationary phase be on beads, you can just coat the surface of your tube with either a liquid that's stuck there or some sort of solid that your analyte's going to interact with. All right, now with that, let's get into the specifics of the different types of chromatography. We will start with adsorption chromatography. So in adsorption chromatography, the stationary phase is a solid, and your analyte will come through and adsorb to the surface. So adsorb here is spelled with a D. It's not absorb, which is spelled with a B. Adsorb with a D is when things just stick to the surface but don't penetrate the surface. Absorption is when things penetrate the surface and go into the interior. So in adsorption with a D chromatography, the analytes or the solutes have some sort of affinity for the surface of the stationary phase. And so as your gas or liquid mobile phase passes over the stationary phase, your analyte is going to have some desire to stick to the surface, but also some desire to be dissolved in the liquid or mobile phase. And so how much it desires those two things is going to affect how long it takes to move through your column. So if you have an analyte that doesn't really care for the surface at all, doesn't want to absorb to it at all, but really likes to be dissolved in the mobile phase, it'll go through the column relatively quickly. On the reverse side, if you have something that loves to absorb to the solid stationary phase, but is not very soluble in the liquid or gaseous mobile phase, it's going to take a long time to get through that column. And so obviously there's a spectrum there where things have different affinities for the surface, different affinities for the mobile phase. And so because of that spectrum in affinities, you can get separation. An example of adsorption chromatography is one that we'll actually be using in Lab 7. In Lab 7, we'll be doing what's called thin layer chromatography. And in thin layer chromatography, or TLC for short, you have an aluminum plate and it is covered in a coating of silica. And that silica is our stationary phase. So then what you do is you just draw a line at the bottom of this plate indicating where you're going to start your samples. You put some labels here indicating which samples are where. And usually if you use a number 2 pencil, the ink won't, or the lead won't be affected by the experiment. But then you'll take your analytes and you'll spot it onto this TLC plate, and that's indicated here with these different colors that I've put here. And so you'll put your different samples in different places, and then what you'll do is you'll take this whole plate and you'll put it in a developing tank, which is basically just a narrow flat tank that'll hold the TLC plate upright. And you'll just put this bottom part below the line in some sort of liquid stationary phase. That's what we call developing the plate. In the stationary or the mobile phase, the liquid will be pulled up through the plate through capillary action. So it'll just have some desire to interact with that plate. So it'll get pulled up, and as it gets pulled up, it will pull up the analytes with it. And then we can identify different analytes by how far they move up this plate. And we usually calculate what's called a retention factor here. And so the way we do that is we start at the bottom where our analytes started, so this bottom pencil line, and we mark the top of this TLC plate where the solvent reaches. So that distance the solvent travels from the starting line up to its finishing point, we put on the denominator of this fraction. So that's the distance the solvent traveled. And then on top, we take the distance that our analyte traveled, and that goes on the numerator. So you literally usually just measure these with rulers. So this isn't a high precision experiment, but it's used all the time in organic labs for separating compounds, and it's a super important technique, actually. Another form of adsorption chromatography is one that I use in my lab all the time. This is called high performance liquid chromatography, or HPLC for short. And we'll get into how HPLC works specifically later on. Here is an old HPLC instrument that I think has been retired now. And in this instrument, we have these columns, and these columns are actually only about 10 to 12 inches long. Actually, in my instrument that I have in my lab, we have some that are only about 8 inches long. And so the analyte comes into one end of this and gets pushed through with a high pressure solvent, and it moves really slow through here. Sometimes can take 2 or 3 hours to make it through a really short column. And as their analyte comes out, we have some sort of detector that detects them. And so right here, this is a spectrum or a chromatogram collected from an HPLC instrument. And you can see each of these peaks corresponds to a different analyte coming out. And so we have some of these species numbered, and the key is right over here, but this one had some benzoic acid and some caffeine and various other things in it. So HPLC is a very powerful separation technique, and it actually gets us pretty good reproducibility in terms of signal heights and so forth, so you can use it for a lot of quantitation as well. All right, the next type of chromatography we'll be talking about is partition chromatography. Partition chromatography is actually very similar to adsorption chromatography. The only difference is that in partition chromatography, you have a liquid stationary phase instead of a solid stationary phase. And so instead of adsorbing to a surface, your analyte is going to spend some amount of time dissolved in the stationary phase. So it's going to have some affinity or some desire to dissolve in your stationary phase and some desire to dissolve in your mobile phase. And so your stationary phase is often supported on some sort of solid. So let's say you have a bead in there or some sort of rugged, jagged stationary phase to give you a little more surface area for your analytes to interact with. Now in this type of chromatography, your mobile phase can again be a liquid or a gas, as long as it's some sort of fluid to flow through your column. And then your analytes partition or spend different amounts of time in the mobile and stationary phase. And so the analyte's relative solubility in the mobile phase versus the stationary phase is what controls how long it takes to move through the chromatography column. An example of partition chromatography is gas chromatography, or GC. So in this instance, you can have an open tubular column that is coated with a liquid surface. This is actually only partitioning chromatography. So GC is only partitioning chromatography when you have a liquid coated surface in there. And then your analytes will dissolve in your mobile phase and your stationary phase for different amounts of time. So if you're looking at these two analytes here, the yellow one and the blue one, you'll notice that the yellow one spends more time in the mobile phase, the blue one spends more time in the stationary phase. So which of these two is going to elute or come out of our column more quickly? Go ahead and pause the video and think about that for a second. All right, well, the yellow species is spending more time in the mobile phase. The mobile phase is what's traveling through the column. And so this yellow species is going to travel through this column more quickly than this blue species, because the blue species is spending so much time stuck in the stationary phase and is not moving. So your relative solubilities then control your elution rate and how well you're separating different species. Now, a note about gas chromatography. So GC is not a partitioning technique when you're using a solid stationary phase. In that case, it's adsorption chromatography. So GC can be either partitioning or adsorption chromatography, depending on the type of column you're using. And of course, you'll choose the type of column depending on what types of analytes you're trying to separate. All right, our next type of chromatography that we're going to talk about is what's called ion exchange chromatography. So what you have in ion exchange chromatography is you have some sort of charge carrier adhered to a solid stationary phase. So if this was a positive charge carrier attached to this stationary phase, you could have NH3, so that would be NH3 plus covalently bound to the surface. If it was a negative charge carrier on the surface, you could have CO2 groups stuck onto here. And that would give you CO2 minus. So this stationary phase right here is actually what is used in anion exchange. So this has cations stuck here, but you're exchanging anions. So when you start, the way this works is you have your cations attached to the surface, and then those will be complexed with some sort of anion on the stationary phase. And as you flow your analyte through, if you have a different anion that likes to stick to these positive charge carriers more, it'll come in and displace the other negative charge carriers. It'll pop that negative charge carrier into solution, and that negative charge carrier will be carried away. And we will review that in the lecture for Lab 8. So glass is actually often used for this type of thing, or at least glass beads are often used for this type of thing, where you have a bunch of O- groups sticking out into the solution. So that's really good for cation exchange chromatography. But some examples of this type of chromatography is actually a Brita filter. So Brita filters exchange ions to remove unpleasant ions from your water and replace them usually with sodium or potassium. If you have a soft water generator, that's operating on the same mechanism. And again, we'll talk about this a little more in Lab 8. All right, let's move on to our fourth type of chromatography, which is size or molecular exclusion chromatography. In this type of chromatography, if you haven't guessed, we're going to be separating our analytes by their physical size. You might think that in size exclusion chromatography, that the little guys come out first and the larger guys take longer, but it's actually the reverse. In size exclusion chromatography, your bigger analytes are actually going to move through your column more quickly than your little analytes. The reason for this is because in size exclusion chromatography, our stationary phase has a bunch of pores in it, and your solvent's going to be moving through those pores. Your larger species won't fit through those pores, and so they will just take the simplest path to get through this column. Your smaller analytes, though, will try and sample as many different pathways as possible, and so they will travel a farther distance, actually, as they travel through this column. So they will try and wind us through as many of these spots as is statistically reasonable, and we will separate our two species out. So what I have here on this plot right here is just time with some peaks that have been drawn by hand here. And so in this, though, since time 0 is over here, the species that's going to come out first is actually this one. And this is a principle that's actually kind of confusing for beginners in chromatography, is that the peak that is seen first is actually the one that's coming out first. So if I give you an actual picture of this type of separation with a pink and yellow analyte, you'll notice that the pink one is coming out first. That means it's bigger. It's going to come out and reach the test tubes first. And so then if we're doing this by hand, we can elute until this whole pink drop comes out, and then we can move over to a different test tube and keep eluting until this yellow drop comes out, or this yellow spot comes out. And then we've just separated our pink and yellow species. So this is actually used all the time for rough fractionations of proteins and to separate things by approximate molecular weight, which is useful for a variety of experiments in biochemistry. And if you want to learn more about that, feel free to ask me. I'll be happy to tell. But here is a picture of a size exclusion chromatography stationary phase. You'll notice it looks a lot like a sponge. It is more rigid than a sponge. It's not so squishy. But what you'll notice is in this sponge-like material, it's got a bunch of different pore sizes. So there's some that are really big, some that are a lot smaller. And because of that, you can separate a lot of different sizes, not just one size that doesn't fit, one size that does fit. What you have is some intermediate species that fit through some of the holes and not through others. And so you're able to separate out a lot of different sizes with an anhomogeneous sponge-like material like this. All right, let's move on to our final type of chromatography for the day. This is affinity chromatography. So affinity chromatography is actually the most selective type of chromatography. In affinity chromatography, we're going to look for very specific binding partners, whether this is a protein or a drug or some other chemical species. We'll design our stationary phase with some sort of structural form that only attaches or binds to one species and lets all the other junk flow through. So the most common example of how this is done is by attaching antibodies to your stationary phase. So antibodies have this general structure where they generally have 6 protein domains. So those are illustrated by the different colors here. And we write that in shorthand looking like this, where we basically have 6 rectangles drawn together with some attaching lines in there. And all these squiggly lines or clear boxes are just different codes that biochemists sometimes use to identify different antibodies and different protein components of the antibody. You can switch out different proteins here and that'll make this antibody stick to different species. But the way that this works is you get a column, you flow through your antibody, you have some sort of reaction that causes it to covalently stick to the surface of your column here. So now you got a bunch of antibodies stuck to the stationary phase. Then you're going to flow your analyte through. You're going to have a bunch of species that do not care about these antibodies at all. So they'll just keep flowing on down. But then if we have the species we're looking for flow through, it's going to come and stick to these antibodies really selectively. So this is really good, for example, if you're looking for really low abundance things. So for example, if you're looking for cancer biomarkers, those are often low abundance. And so you need to separate those out from the big old mess of really abundant things in your blood or serum or other things that you take out of your body. So once the analyte is done washing through though, you'll often actually get some other species stuck on your column, usually due to steric effects. So something might get stuck in between 2 of the antibodies, or it might have a really, really low affinity to it and just happen to have one molecule or a few molecules get stuck on the surface. So usually what we'll do is send a wash through, get anything that might have gotten stuck in there that's not supposed to be stuck out of there. And then what we want to do is detach our analytes of interest from this column so we can actually use them for whatever we want. So right now these green stars are stuck to the sides of our column. So we're going to send through a releasing solution that'll cause them now to fall off and come out. So typical examples of these releasing solutions are solutions that change the pH, so either make it more acidic or more basic, so you change whether your antibody or your protein or whatever you're looking for either gets protonated or deprotonated, or you can change the salt solution in here. It's going to be something that either changes the chemical properties of your protein or causes your protein to change shape and wash out of it. But now you have a really pure sample of your protein of interest. All right, so let's review the types of chromatography we talked about. So the first was adsorption chromatography. In adsorption chromatography, you have a solid stationary phase. Your mobile phase is a liquid or a gas. Your analytes will adsorb to the stationary phase's surface. They won't absorb into its interior. And the analytes affinities for the mobile and the stationary phase control the rate of separation. Next type of chromatography was partition. Partition chromatography, you have a liquid stationary phase and a liquid or gas mobile phase. Your analytes dissolve in the mobile and stationary phases, and how much the analyte wants to dissolve in those 2 phases controls the amount of separation you get. An ion exchange chromatography on a solid stationary phase that has ions attached to it. You have a liquid mobile phase, and your analytes undergo ionic bonding with your stationary phase, releasing some other ion that was bonded to the stationary phase. In size and molecular exclusion chromatography, you again have a stationary phase that is a solid, the mobile phase is liquid. Your analytes separate by their size, by their ability to fit through different pores. So they separate basically by which pathways are available to them as they travel through the column. The last type of chromatography we talked about was affinity. This has a solid stationary phase with a liquid mobile phase, and use antibodies to isolate a single species at a time to get really high selectivity. And that is our introduction for chromatography. Let me know if you have any questions about that. I'll be happy to